Okay, then we start. So the last lecture of today is on data integration of spatial data. So just to make sure that we are all on the same page, I think if you're doing analysis, I think what we have covered so far is a little bit on the image processing or pre-processing of the data, a bit on QC, and normalization, filtering, and so on. So everything from now on depends on actually what you want to do with the data. So to me, that is more downstream analysis. So these are not things that you will have to do on the data. It really depends on the question that you want to ask. So I put here sort of examples of questions that you, or types of analysis that you might want to do. So you might want to annotate your data. You might want to identify spatial clusters or domains, or look at cell-cell communication. These are all things that we'll cover tomorrow and the day after. But you might also want to do deconvolution. If your data is not single cell resolution, you might want to predict genes that you haven't measured. Or you might want to align multiple sections and so on. So these last 3 points are the points that we will cover briefly in this lecture. There might also be other things that are not on this list as well. So I thought a bit about integration, and I think I would like to talk a bit about single cell and spatial data integration. What can we do with that? And what are the different approaches and methods? A bit on alignment. So I mentioned briefly batch correction, but that doesn't take any space into account. Alignment can be of consecutive sections, and that can also be a bit, you can see it as a way of doing batch correction. And finally, very briefly on multi-omics, if we have multiple measurements of different technologies. Okay, so why would we need to do integration across single cells and spatial? I think the reason is that every technology gives you sort of a different picture or a different image. So with single cell data, you're usually untargeted, and that's between quotes, because it's untargeted as long as you're interested in poly A RNA species. If you're interested in other things, then you consider it targeted, right? You're only fishing polyadenylated RNAs. But you have no spatial information. If you do sequencing based, as we saw, then you're again doing sequencing, so between quotes, untargeted, so you get the full transcriptome. But the resolution is, in general, low. Of course, some of the newer approaches are much higher resolution, but you still suffer a bit. For example, with Visium HD, if you go to the 2 micrometer resolution, then the data is very, very sparse. So most people would like to use it at a higher resolution than the number of cells, for example. This might improve in the coming years. And the last one is if you use in situ sequencing or fish-based approaches, then your resolution is very high, sensitivity is high in general, but usually it is a targeted approach. Of course, there is a more complex picture, as Lars showed you yesterday, but this is largely the main limitations. So in this case, you might want to integrate your spatial data with single cell data. And with that, you can have multiple aims in mind, right? So at the top, for example, you can sort of focus on embedding your cells from the single cell data into space, right? So find their locations. And if you do that, then you can do deconvolution if this data is not at the single cell resolution. Or you can just better understand the context or the spatial context of your dissociated cells. Yeah. The other point of view is if you're more focused on the spatial domain, that you can also think about predicting genes that you haven't measured, right? So you borrow information from the single cell data to predict or impute how these genes would look like. Would you have measured them in space? Yeah. These are sort of, at least my own classification of the types of methods that are used to do these tasks. And what they can do, right? So you can do joint embedding of the 2 data sets, right? So you try to learn a latent representation of the spatial and the single cell data. And if you do that, you can do both tasks, right? So then you can look for any spatial cell at the closest single cell data and then borrow gene expression information from there. Or you can do deconvolution, right? Of the spatial spots. You can do some modeling of the gene expression that you observe in space, right? Given profiles that you learn of cell types from the single cell data. And with that, you can only do deconvolution, right? So it's very difficult then to do imputation because you don't have that much information to borrow from the single cell data. And then you can think about directly mapping. So for every single cell, you sort of try to predict what's the most likely location of that cell in the tissue. And there are multiple approaches to do this. So what I will do today is that I picked one example of each category. So Spage is our own tool, so I'll explain that because it's easier for me. For probabilistic modeling, we picked cell to location to have a look at. And for the mapping, we will talk about Tangram. I do find these approaches, so both NovoSpark and SPA-OTCS, they use optimal transport, which I think is a very interesting concept and very useful in this case, but we will not cover it today. But I really encourage you to look a bit more into that as well if you're interested. Okay, we'll go backwards, right, from Tangram to cell to location. There, for both methods, I will not show results. I'll show some results from our own tool and a bit of benchmarking, so you know at least what we're going to cover. Okay, so starting with Tangram. So Tangram is trying to basically learn a mapping of your single cells to spatial locations. So you start basically with two matrices, right? You have S, which is your single cell data, says cell by gene matrix, and G is your spatial data. So you have voxels or locations by gene. Yeah. So what you're trying to do is to get a mapping. So the dimensionality of this mapping matrix is cell by voxel or cell by location, right? So then it will tell you what's the most likely location of each individual cell. And they want it to be a probabilistic mapping. So that means that the elements of this M mapping matrix is larger or equal to 0. And it really gives you the probability of this cell I being mapped to a particular voxel or a location J. Yeah. And then again, to make it a probability, then the summation for every cell over all the locations equals 1. Yeah. So then maybe you have very high probability to map this cell to a particular location and then low everywhere else, or it's uniform if you don't know, and so on. If you multiply the transpose of this mapping matrix by S, then you get the predicted gene expression, right? Because it's the mapping of the single cells to locations. So this multiplication gives you the predicted gene expression, right, of spatial locations. And in the locations, you have this vector mj, right? And mj is basically the division of this mapping probability by the total number of cells. So you can see this for every voxel as the sort of the density of the cells at this voxel. Yeah. So it's basically, do I have high probability for many cells to map to this particular location, or only one cell has a high probability to be mapped to this location? Yeah. Okay, so how do we learn this mapping M? They construct an objective function that they want to minimize, right? So we'll go through this objective function. So the first term in it is a KL divergence, right? So the difference between M, this vector over here, so that is the density of cells at every location, right, that we are predicting, and the actual density of cells. So if we're working with single cell resolution data, this actual density of cells is just a uniform distribution. So for every location, we have 1 over the total number of cells in the tissue. If we're working with the Visium data, for example, this density is the number of cells in every spot. Yeah? And you can get this estimate by segmenting the H&E image that comes with the Visium. So you segment the nuclei, and then you say, well, I have 5 nuclei below this spot, and I have 2 nuclei below that other spot, for example. Yeah. So using that, you get a density estimate of cells in your spatial tissue. Okay, clear? Okay. And then you have another term that gives you that you actually want to maximize, and that's the cosine similarity between the mapped or predicted spatial expression and the actual spatial expression. And this matrix is now sliced over the genes, right? So you take one gene and you look at the predicted expression pattern over the tissue, and you have the measured over the tissue, and you want those to be similar per gene. Yeah. And then you loop over all the genes. This term is very similar, but then it's flipped. You slice the matrix differently. So here you actually want the expression of every location, the actual expression that you measured, and the predicted ones to be similar. Yeah. So you want the expression profile of every location or voxel between predicted and actual to be similar, and you want the expression of each gene over the tissue to be similar again between predicted and actual. Okay. So 3 terms: density, expression of a particular gene, expression of every voxel. Yeah. Okay, in practice, this is the objective function. Looks a little bit more complex. So you have the same 3 terms that I explained over here, plus additional things. These additional things are also quite easy. So this is an entropy over the mapping. So you want your mapping to have high entropy between the values that you're learning. So you don't want to have the same value everywhere in the mapping, because otherwise it's useless, right? And then you have this F over here, which is just a filter, right? So you want to allow your model to say that some cells I don't want to map, right? So if you have a mismatch between the number of cells in the single cell and the number of cells in the spatial data, you want to be able to say, well, these cells I'm not going to map, or these locations are going to be empty in a way. Yeah. So they do this with this filter matrix, and these are just matching the number of mapped cells to the actual targets in the spatial data, and enforcing that these mappings are Boolean, so they are closer to 0 or 1. Yeah. But okay, details are not very important. It's just a filter to allow you to have mappings between data sets that have different numbers of cells. Okay. If the modeling part is clear, so what can you do actually once you have done this model? You can look at, they call it extend gene throughput. That basically means predict the spatial expression of genes that you haven't measured, right? Because now I have mapped every cell to a location. So I can just borrow the expression of that cell from the single cell data or single nuclei data. You can localize cells. So if you want to know what's the most likely location of a particular cell, you get that by the probability, the mapping probability. You can do deconvolution. So if you have mapped to Visium data, you can then look, okay, I mapped here, I don't know, 50% of the cells mapped to a spot are of cell type A and the others are cell type B. So then you end up doing deconvolution of the signal in that spot, and so on. Okay, let's go to cell to location. And again, very briefly explaining what the model is trying to do. So the cell to location is really trying to model the spatial counts that we get, right? And then they start again from a simple assumption. This is a negative binomial distribution of the counts. So you have a mean over every location and the gene, right? And then you have a unexplained variance that is per gene for each batch. And the batch here you can see as a different slide or experiment or, yeah. For the sake of this, at least explanation, you can ignore the batch information for a little bit. So what is happening? So now we try to model this mean expression of a gene at a particular location in the tissue. And then we do that with this formula. So the core of it is relatively simple, right? So for every location in our tissue, again, assume it's a Visium, for example, spot, we have a mixture. So these are weights that we learn for every cell type. And these are the average expression of that cell type that we get from the single cell data. Yeah? So you have your single cell data that's labeled or clustered into different cell types. You have the average of every cell type that goes into here. So F is the number of cell types that you have. G is the number of genes. And then again, you're learning a weight for every spatial location for every cell type that you have. Yeah. So eventually, this is the outcome of your deconvolution that you want to learn. Yeah. What is the contribution of every cell type to the expression of the spot that I'm interested in? Yeah. Most deconvolution methods that rely on non-negative matrix factorization and so on work a little bit in that framework, right? That they want to decompose the signal in one location into a linear combination of signal from different cell types. There are a few things additional to the model. So here you, before we move on. So the main contribution in cell to location is that they do use a prior on this weight matrix to model autocorrelation, right? So if I estimate this independently for every location, it might be that I get, I don't know, 90% neurons in this spot, and the spot nearby I have only 5% neurons. That's unlikely in spatial data. So there is again autocorrelation, there is spatial dependency to a large extent. So if you predict a certain proportion of cells in one location, your expectation is that the nearby spot will have a similar, or there is a continuous change in the proportion of cell types. Yeah. And it depends on how harshly you enforce this, you allow sort of abrupt changes in the tissue in terms of composition. Yeah. This is a term to account for the fact that here we're talking about gene expression profile of cell types that come from single cell data, but we try to use it to explain gene expression that we measure in space. Yeah? And these are two different technologies that will have different sensitivities, for example. So this is a term just to account for this. Yeah? Here you have this is an additive shift. So if you look at it, it's per batch, and then, so ignore that, and then it's a gene-specific factor. So this is just to account for any background noise across the entire section that you're looking at. Sorry, this is to account for differences across these batches, right? So if I have differences in measurements across the batches, and this term is to correct for the differences across the tissue itself. Okay. Slightly more complex model, but at the end, we're trying to learn these weights that tell us, okay, in this particular spot, what is the weight or the contribution of every cell type that I have from my single cell data to the observed count over there, Nicholas. Okay. This is our own method, and it's in the class of trying to learn a joint embedding. So just to revisit, in this particular case, we focused on predicting genes that you haven't measured, but you can also use it for other things. So again, you have two data sets, one spatial data set, single cell level resolution. You haven't measured all the genes, you have measured the subset of the genes, and you want to borrow from a single cell data to predict the rest of the genes. So the way we do this is that, okay, you take a spatial cell in which you have measured a certain number of genes. You look at all the cells in the single cell data, and using that overlapping set of genes, you look for the nearest neighbors. And then you can impute the genes that you haven't measured based on these nearest neighbors. And you can do KNN imputation for this. So that sounds quite easy, but then you get to the problem where these data sets live in completely different spaces. Yeah. So what I'm showing you here is the cosine similarity between the principal components of the spatial data and the principal components of the single cell data. And what you see here is that the cosine similarity is in general quite low, right? So these data sets are completely not aligned in a way. So if we now use the trick that I just showed with the KNN imputation, we will get garbage, right? The distances will not mean anything in a way. So we need to correct for this in a way. And the way we do it is with an alignment approach called precise. And the way precise works is, again, relatively simple. You have principal component analysis done on the target data sets. So in this case, I think target is the single cell data. And you do principal component analysis on the query data set. So these are, again, the principal components I showed you, but now you want to align them. So what you can do is that you can take the multiplication of the two matrices that gives you the cosine similarity. And if you do singular value decomposition on this matrix, then you end up rotating one data set to the other. Yeah. So what happens, again, you have these principal components here from spatial, principal components from single cell data. And if you do this, then what you actually end up doing, you call them now principal vectors. You try to define the PCs that have the maximum similarity, sort of. They're now called principal vectors. So PV1 is now the pair of principal vectors that has the maximum similarity between the two data sets. PV2 is the one with the second highest cosine similarity, and so on. So in a way, you're rotating one space to align with the other one. And if you now look at this cosine similarity matrix, no surprise, it's very diagonal and ordered, right? So now the first pair of principal vectors has the maximum cosine similarity of 0.78, for example, and then it keeps going down over here. And now this is an aligned space, right? So we can now measure distances and do KNN imputation and so on. If you use SRT, for example, it is doing the same, but then instead of the precise method that we're using, they're using canonical correlation analysis. If you use Liger, then it's again the same, but with an NMF approach and so on. Some methods merge the two things. So here we do alignment and then prediction. But if you do your embedding with something like SCVI, for example, then you're not really doing this KNN imputation. You can just sample from the corrected latent representation directly. Okay, just to show you some results, because so far we haven't looked at results. So this is primary visual cortex data. So with star maps, so here they have measured around 1,000 genes. And this is single cell data from the Allen Atlas of the same region, where they measured the entire transcriptome. Okay. So how do you evaluate this? You take a gene that we have measured. I apologize, this is a flipped version of this one. So the cortical layers are ordered this way vertically now. You take a gene that we have measured, you remove it, you apply the whole procedure, you predict the expression of that gene, and then you look at the prediction pattern, and then it looks quite okay. And then you can do this in a leave one gene out manner. So you leave each gene out one time, and then you predict it, and then you look at the correlation or the similarity of these results. And in this case, Spage seems to work quite well, even if you have very sparse pattern over here. But that's what all the methods are doing. So how well do they do? There are two large benchmarks. So this one looked only at deconvolution, right? So they compared all the methods that could do deconvolution. Of course, that's always the risk with these benchmarks, is that people claim, okay, the result is in the abstract that there are three methods, that is card, cell-to-location, Tangram, that works the best. But of course, if you read the paper, it's a little bit more complex than this. But okay, these are the three methods that are highlighted here. Same goes for this paper. So here they did both deconvolution and prediction or imputation. So predicting genes that you haven't measured in space. And in this case, Tangram, GimVI, and Spage did very well. And in terms of deconvolution, I think it was cell-to-location, spatial DWLS, and RCTD. Yeah, so you already for deconvolution, you see there's one overlapping in the top three over here. That's cell-to-location. But yeah, again, if you are interested in this work, you should read the papers. This is a very simplified, naive way to report the results. I added this because, again, Spage here did very well. So we're also very happy with this. So this is a paper particularly, I think Naveed was also involved in this one, particularly on Xenium data. So these are this is, for example, the ground truth, and these are all the predictions of all the methods. And they have 4 metrics here to evaluate how well do methods do. And I think Spage, for example, did well on like 3 of the 4. It's the top one. And yeah, but if you look visually at the results, they are actually very similar, right? So, I mean, the differences between the methods are also not that large. Yeah. There is more into these benchmarks also in terms of the computational power that you need and the RAM and so on. Also quite important if you would, for example, try to run the Murphish Atlas from the Allen and try to predict all the missing genes over there, then it becomes quite problematic for a lot of these methods. Okay, is there any question on the single cell spatial data integration? So for the practical part, we will only do stuff related to the previous thing. So we will try cell-to-location for deconvolution. We'll do Tangram and Spage for prediction of missing genes and comparing them. Yeah. But I also thought let's cover other topics. So they're not in the practical, but at least you have a bit of an idea on them. So the first one is about data alignment. Some people call this pseudo 3D or virtual blocks, or I don't know what other name come up with in the coming years. But the idea is simple, that you have a tissue and you're interested in the 3D organization of the tissue. Most of the spatial protocols that we have are 2-dimensional. Right? So you can just do consecutive sections or multiple sections. So here, for example, you can do 90 slices and whatever. And then the aim is that you sort of align them into one 3-dimensional volume at the end. There are multiple methods already doing this. I split them here into alignment and integration. That split is also a bit shaky, right? So it helps in explaining, but it's not fully correct. So the integration ones are a bit similar to what you would do with the single cell data, right? You're trying to sort of do batch correction. You're trying to learn a joint representation of all the sections that you have, yeah? And in that process, you're also sort of aligning them. But the other one is sort of more similar to image analysis, where you're trying to just warp the images or the gene expression measurements that you have so that they align without learning a latent representation of those. Yeah. I'll try to go through a couple of examples so you get an idea what they are trying to do. One of those is called paste. So what paste is trying to do, so again, you have your tissue, and you have taken multiple slices, and in each case, you have the coordinates of the spots or the locations, and you have the gene expression matrix that associates each of these spots to a gene expression vector. So paste is based on optimal transport again, and that's why I said it's a very nice or a hot topic now because you can use it for multiple things. So what paste is trying to do is that again to learn a mapping between between spots from a query to a reference, for example, right? So what's the best match for this spot in this section? They also have a mode where you can, if you have multiple, that you learn a central alignment of your tissue, but they are very similar. So let's just explain one of them, and I think the other one is you can read in the paper. Again, it's optimal transport based. So what optimal transport gives you at the end is a mapping, right? What is a mapping? It's just a matrix that says, these are all the locations in section 1. These are all the locations in section 2. What's the transport map, right? What is the most likely mapping of this spot in section 1 to all the other spots in section 2? Yeah. So these mappings are the pi over here. So I and j will be two spots on two different sections. So they will be this I and j, for example. And what you're trying to optimize in this function is the similarity between the expression of the mapped spots. So if your mapping at the end says I and j matches, then the expression profile of these two spots or locations should be similar, right? It should not be, or it might not be identical, but it should be very similar. Yeah? And the other thing is the spatial distance preservation. It's basically saying, okay, if I have a mapping from I to j and a mapping from k to l, then the distance between ik over here should be proportional to the distance between j and l over here. Yeah? Okay. So if you optimize this thing, and I don't know how you optimize this thing actually to get these mappings, then you end up with the sort of the best possible mapping that maximizes gene expression similarity between matching spots and preserves distances between pairs of spots on the two sections. Yeah. Okay. Other methods use different approaches. So cast, for example, is a method that if you have multiple slices, right, you have the location and you have the gene expression matrix associated with these locations. What you do is that you build a graph of every section. So the nodes on this graph are the spatial locations, and the connections are the spatial distances between these locations. And you have these sort of node features, these small vectors over here. This is the gene expression profile of every spot or location. Yeah. And then you use a graph neural network to learn an embedding of these graphs. Yeah. And you do that jointly on all the sections that you want to align. So what effectively happens here is that you get a representation in which two spots will be close by if the graph structure of them is similar, and also that their feature vector is also similar. Yeah? So these spots are similar in the two sections. They will have similar neighborhoods, and they will have as similar as possible gene expression profile. So they will end up in that embedding close by to each other. Yeah? And then you can use that information to warp one image to the other one. Yeah. Okay. Can skip ST aligner. It's very similar approach. People are trying to build frameworks around this, right, to build 3D. So this is one from Nikolaus Raevski's lab where they try to get these different sections on a tissue block and then try to align them with their own method. It's called STIM. So STIM basically says, well, if I have two sections over here, I can look at one gene, right? So one gene in the two sections. So that gives you an image, sort of. You can render the gene expression as an image. And then they do two things. They extract features from the image with SIFT. That's a very classical computer vision approach to get image features. And then you can match them with something called RNA-seq. Yeah? So then you take one gene that gives you two images from the two consecutive sections, and then you can align them. But of course, you have multiple genes, so you can try to solve this sort of mapping problem globally across all the genes that you have measured. You can have a look at STIM. It also allows more things about rendering in 3D and so on. So, and they give one example. It's an example on cancer, which I know nothing about, but what they try to explain here is that if you take one 2D section, there might be a structure over here that you sort of say, well, this is too far away from here, for example. While in 3D, these two things might be connected, right? That you have to look at a different depth to sort of link these two features that in one two-dimensional image are too far away. Yeah. I think there should be a little bit more things done to convince people that three-dimensional relations, like for example, building graphs in 3D and showing that the patterns you get from 3D graphs are really unique compared to the ones in multiple two-dimensional graphs and so on. But I think there will be a lot more of this in the coming. Again, as always, there is a benchmark paper comparing a bunch of these methods. So they do also clustering, so that will be discussed tomorrow. But they do compare multiple alignment tools and their ability also to do batch correction. This part we haven't discussed, so I didn't include anything on this. So these are all methods that try to learn a low-dimensional representation of your data, so similar to SCVI or CRAT with the canonical correlation analysis and so on. But they do include the spatial information in that learned feature, latent representation. Yeah. But yeah, I haven't had time to look at those, and I haven't seen them being used beyond their own papers. These are all very recent ones. Very few words about multi-omics. So of course, many people are interested in things beyond transcriptomics, right? So you might want to measure proteomics or metabolomics or other omics features on the tissues that you're interested in. So there are sort of two ways to do this. I think the hard way, this is an example from Leiden, where we tried to have mass spec imaging and imaging mass cytometry on the same section, right? So you have the tissue section, you do MALDI, so you have this matrix on top, and then you do laser excitation, so you get a mass spec, and then you remove that matrix, and then you do mass cytometry. Yeah, and the idea is that mass cytometry will give you the cell types, and the imaging mass spec will give you lipidomics and metabolomic measurements. Yeah? It's on the same section, so the alignment problem is actually quite easy, right? Because it's exactly the same tissue section. One of them is a bit smaller than the other, so you have to crop a little bit, and then the resolution is different, so you have to do something, downsample one modality to the other, and so on, but all very simple procedures. And what you get at the end is something like this. So again, you can cluster based on your mass cytometry information into different cell types, and these are all features that you can get from the imaging mass spec. And here is an example. Again, I think this is a colorectal cancer sample where you have the tumor over here, and in the middle you have the immune stromal compartment, and then you can look for features from the imaging mass spec data that are different between the two compartments. Yeah. Then you get information from both domains or both data types. But of course, in practice, it's very difficult to do this, right? So already, I think many people are trying to combine transcriptomics with many of these technologies, and then RNA quality drops, for example, if you try to do imaging mass spec and so on. I think it will be solved eventually, but we are always interested in many more layers that might be difficult to combine. So an approach to overcome this is to do two consecutive sections from the same biopsy that you're interested in, and then you use image registration to align both. So if you have done this, then you have correspondence or matching between spots or cells in the two sections. And then you can turn it into a vertical integration problem, right? So now for the same location or the same matching location, I have information from modality 1, and I have information from modality 2 and maybe 3 and so on. And this you can solve with any of these vertical integration methods like MOFA Plus or CCA, Jive, Total VI, and so on. Yeah, where you can learn an embedding using two sets of features instead of one. Yeah, but you're anchoring yourself on a cell or a spot, and that anchor you get by the mapping of these spots between the two sections. Okay, just to give you an example of, okay, how can this work? There's a method called ELD, effortless landmark detection. And the way they do this is that you have your two sections. You do landmark detection separately for each section, right? There are tools to do this. So you get automatically identified landmarks. And then you can use existing methods to do landmark-based registration. So these tools will try to find the best matching between the landmarks sets that you identified in both sections. If you do that, then these two are aligned, and then you can do this, basically, yeah? Because then my tissue is now aligned between the two sections, and I can get features from modality 1 in section 1, features from modality 2 in section 2, and then do whatever you want with that.